Hi everyone. Hope you did well on test number 4. I just finished grading those, and now I'm going to be talking about photosynthesis. Now the ones in the evening class will be taking test 4 this Monday. But regardless of what class you are in, you are about to be wowed and amazed, and probably a little intimidated, to be quite honest, because photosynthesis is kind of a complex process, but fortunately, it's just the opposite of the one that you just learned about, cellular aerobic respiration. So we'll be looking at our key concepts, an overview of photosynthesis. We'll be looking at those reactions that harness light energy called the light reactions. We'll look a little bit at the molecular features of photosystems. Photosystem 2 is the first one, and photosystem 1 is the second one, because photosystem 2 was discovered first, and they involved electron transport chains. Then we'll look at synthesizing carbohydrates by way of the Calvin cycle. All this is happening in the chloroplast. And then lastly, we'll talk about variations in photosynthesis when we go through this whole process. So just hang on to your hats. Hang on to your leaves. Get a nice crunchy, juicy, sweet apple while we're talking about it, and think about all those great sugars that are produced, because that's what happens in photosynthesis. That reaction of that you just wrote down for your test, hopefully you did, and some of you might not have gotten all the particulars, but it's the same exact reaction. As a matter of fact, it takes 6 CO2s. This equation, I don't like that because it's not balanced. 6 H2Os, light energy, and it yields C6 H12O6, which is glucose, plus 6 O2s, plus 6 H2O. So you have that, and that balances that equation. So in this process, carbon dioxide is reduced. It is reduced to C6H12O6. Water, or H2O, is oxidized. Oh, my goodness. Really, you know what? It should say CO2 plus H2O plus light energy yields C6H12O6 plus O2. That's that third H2O on there should not, or that second H2O on as the one of the products should not be there. It's really, it uses up water, and water is oxidized into oxygen, right? And energy from light drives this reaction. So let's see if I've got a better slide with the formula on it. Let me see. A balanced formula. I'll have to write it up on the board. I have it on a t-shirt, actually. Yet another nerdy piece of clothing that I own. I'll probably wear it for you. I don't see it in here. It's a shame that it's wrong, the very first one, so I'll have to change that. You know, sometimes with these, they actually put these out as images. Let me see if that's the way this one. I don't think it is, because let's take off that H2O. Yeah, that is an image right there. So I can't change it. Oh well, I'll put it up on the board a little differently. So remember, there's no second H2O. It's just CO2 plus H2O plus light energy, and that's 6 CO2 plus 6 H2O yields 6. Yields 1 glucose molecule and 6 oxygens. Okay, photosynthesis powers the biosphere. So the biosphere is just every place where there's life on the planet. The areas of the Earth, deep in the Earth's crust even, and in the atmosphere of the Earth where living organisms exist. And everything, all of life is driven by photosynthesis. It's the coolest thing. So plants produce the molecules, these organic molecules that cells use for energy. And so plant cells, fungal cells, animal cells, all of those cells are using these organic molecules for energy. Even bacterial cells and archaeal cells, some of them, not all of them, but most of them. In the process, plants are producing oxygen. So when we are talking about photosynthesis, it's a good idea to have a common vocabulary, right? These words are always complicated, so this common vocabulary. One thing that you're going to hear about a lot in the biological sciences are trophic levels. And if you remember when you are talking about energy levels or the way that we acquire energy or food, we talk about whether or not we are autotrophs or heterotrophs. Heterotrophs must gain food and energy from outside of themselves. They must ingest it or absorb it. So they get the organic molecules that provide them with energy outside of themselves. Autotrophs can make the organic molecules themselves from inorganic sources. Some use other sources of energy besides the sunlight. Some use a chemical energy, but most are photoautotrophs. They use light as the source of energy in order to build these organic chemicals. Remember, we're going to start talking about an endergonic reaction where we have to put energy into it in order to build an endergonic anabolic reaction in order to build these glucose molecules, right? So a photoautotroph uses light, and those would be the photosynthesizing organisms like green plants and algae and cyanobacteria that are little bacteria that photosynthesize. This shows the energy cycle. And remember, energy cannot be created or destroyed. It can only change form. And every time it changes form, it dissipates as some dissipates or goes to entropy as heat. So you've got to put light into the process of photosynthesis to generate these organic molecules such as glucose, C6H12O6, plus oxygen. Oxygen is a waste product of photosynthesis, and it's harmful to cells. So the plants need to get rid of it, and they excrete it out. They send it out by way of their stomata. But animals take in those substances, and fungi, animals and fungi, and some bacteria, and plants do too. Plants will too use those substances in their mitochondria for cellular respiration in order to make a lot of ATP. And they break it down into carbon dioxide and water. And that carbon dioxide and water is breathed out or excreted from the animals and fungi and bacteria, and then it's used again in the process of photosynthesis. So you talk about the cycle of life. Tapinya. We'll keep it interesting here. Well, sorry, that's my version of the Lion King, but it's the cycle of life. I wish that I'd really love to see that play again. That's a beautiful, beautiful one. Oh well, okay. Cycle of energy, cycle of life, everything is recycled and used again. Chloroplasts are the little, wonderful, special organelles in plants and algae, photosynthesizing algae, that carry out photosynthesis. And they will use a number of different pigments, but two of their main pigments that they use are chlorophyll A and chlorophyll B. This happens, most of the cells that have the most chloroplasts are within the leaf tissue, and that's called the mesophyll of the leaf. The leaf has clear epidermal cells that the light can shine through, and the light will shine through and hit the mesophyll. And then underneath that thin leaf will be these little openings called stomata that can bring in carbon dioxide from the air. They're just like little pores. Stomata means little mouth. And they bring carbon dioxide into that mesophyll area of the leaf. So the carbon dioxide can come into the stomata and come into the mesophyll and enter into the chloroplast in the mesophyll. And then the oxygen that's produced will leave the chloroplast, leave those cells in the mesophyll, and then leave by way of these pores called stomata. And did I mention that stomata means little mouth? It looks like a little mouth when you look at it under the microscope. So this shows the structure of a leaf. You can see those epidermal cells, and then below those clear epidermal cells, you see the mesophyll cells. And you see the palisade mesophyll cells and the spongy mesophyll. Well, all of those have chloroplasts, lots of chloroplasts in them. And so the light will strike them to fuel the reaction. Remember, photosynthesis, it has to have the fuel for that we have to put energy into this reaction, right? It's an endothermic reaction. So the light energy fuels it, but then the carbon dioxide brings the building blocks, the carbon building blocks that are needed to make glucose and cellulose and starch and sugars and all that sweetness of the apple that you're eating. It all comes from carbon dioxide. And then oxygen is a waste product that leaves by way of the stomata on the underside of leaves. Leaves are very cool structures, little solar cells, or big solar cells even. Okay, so the anatomy of a chloroplast. If you remember when we were talking about cell organelles, chloroplasts are kind of like mitochondria in that they have an inner membrane and an outer membrane, but they're arranged in slightly different ways. The inner membrane is not folded like it is with the mitochondria. They have an inner and outer membrane, and they have a space in between the two called the intermembrane space. But there's also a third membrane in a chloroplast, and that is the thylakoid membrane. And the thylakoid membrane has the pigments like chlorophyll A and B, and other pigments as well, like xanthophylls and carotenes and rhodopsins. And these thylakoid membranes enclose these little sacs, these little special flattened sacs called thylakoids. And those little flattened sacs are stacked up on top of each other into a stacked structure called a granum. And deep within the thylakoid, they look like little fat pancakes. And inside that pancake, there's some gooey stuff, right? Just like there's gooey stuff in the thylakoid. And deep within the thylakoid, it's called the lumen. That's the inner space where the gooey stuff is inside the thylakoid. There's also gooey stuff inside the chloroplast, kind of a fluid-filled area in the chloroplast, where the chloroplast DNA is and everything. And it's the stacks of thylakoids or grana are also in that gooey space. But this gooey space is not within the thylakoid, it's just within the chlorophyll. And this area is called the stroma. That's the stroma. It's the fluid-filled region between the thylakoid membrane and the inner membrane. So it's actually between the innermost membrane of the chloroplast and then the thylakoid membrane. It's the gooey stuff that the thylakoids are sitting in. This picture will probably help you with that. Here's the chloroplast with the outer membrane and the inner membrane and that intermembrane space. And then you see these thylakoids. They look like little flattened pancakes or squishy gummy things stacked on top of each other like in a structure called a granum. And deep inside those thylakoids where the squishy stuff is for the little gummies, that's the thylakoid lumen. And then the gooey liquidy part that the grana are sitting in is inside the inner membrane is called the stroma. And these are all within these mesophyll cells. Some of them will have lots of chloroplasts, and the chloroplasts can actually reproduce on their own through binary fission. They have their own circular piece of DNA, and they're pretty cool. Okay, what's going on in that chloroplast? Why do we care? Well, you should care because it provides you with basically all of your food. You wouldn't have food if it weren't for this beautiful process of photosynthesis. And it's a little bit clumsy. It's not like we think. It's not so graceful and gorgeous, but It's pretty cool. Now, you can separate it into two parts. The first part would be called the light reactions. The light reactions are the way that the chloroplast gathers up light energy and uses that to make ATP, a special intermediate molecule that's kind of like the NADs in cellular respiration, but this time there's a P in it for plant. P for plant. I have to really emphasize that when they're on different tests, when they're on the same test, excuse me. So NADPH, and then oxygen is also produced. In this case, that sweet little grandma at the end of the electron transport chain, we're going to call her Nana P. Nana P. And that's NADP. And she'll be the one accepting the electrons home. Okay? And those electrons use this that pump hydrogen ions, bounce down that electron transport chain and pump hydrogen ions just like they do with the mitochondria. But the hydrogen ions get pumped into the stroma, and then the hydrogen ions rush through ATP synthase. Actually, I think they get pumped into the thylakoid lumen, deep in the inner part of the thylakoid. And there they concentrate, and then they rush through. It's kind of like the money that goes in to the little foster families. And then they rush through that little bit of money, just rushes through that ATP synthase and causes that circus ride to spin and produces ATP. And that happens because of sunlight. That's why it's called the light reactions. And all that process is just there to fuel the next phase. Two phases, remember, light reactions and the Calvin cycle? And Calvin cycle, think this is again a cycle where the last, the first part of the cycle gets regenerated. And for this Calvin cycle, think A for plant. Calvin has an A for plant. This happens in the stroma, outside of the thylakoid, stacks of thylakoids or grana, but still within the chloroplast. And the ATP that's produced in the light reactions and the NADPH that's produced in the light reactions will provide the energy to build a glucose molecule or half of a glucose molecule from carbon dioxide. It's a really cool process. And think of it, it's kind of like cellular respiration in reverse. So the two stages of photosynthesis, the light reactions cause a reaction to happen on the thylakoid membrane, and that it also involves an electron transport chain, but the light causes something to happen in the pigment there in the thylakoid membrane, bounces an electron out, and then the electrons bounce down this electron transport chain and produce ATP from ADP. And then that ATP and NADPH is used to fuel the Calvin cycle where carbon dioxides are attached together. It takes a lot of energy to do that, and they make sugars. Now in the light reactions, another thing happens, you notice over here, water or H2O gets split. Water is split during the light reactions and it gets converted to oxygen. Okay, so what are reactions that harness light energy? What are those? Well, how in the heck can we? It's amazing that plants have been able to do this. Light is a particle and a wave. That's a cool thing about light. We talk a little bit about physics, we talk a little bit about chemistry when we talk about biology because we need them all, right? Light is electromagnetic radiation, and we only see a small portion of the electromagnetic radiation that hits the Earth. We can't see UV light, we can't see infrared, so we just see a small portion of visible light, and that's the electromagnetic radiation that hits the Earth. It's traveling as a wave, and the wavelengths can be either very short or they can be very long. But light also behaves like a particle. They're called photons. And those photons have a lot more energy in the shorter wavelengths. And here's a cool spectrum, the electromagnetic spectrum. And you can see this tiny little place in here where we have visible light. And so what you need to remember is that acronym, Roy G. Biv. That tells you all the colors in the spectrum. So if you split, remember white light is actually a mix of all the colors. So if you split light up into all the different wavelengths of the spectrum using a prism, you'll have red for the first letter of Roy, orange for the second letter, yellow for the third, G is green, Biv means blue, indigo, and violet. So we're going from the red spectrum all the way to the violet. And you see red is 740 nanometers. It's a longer wavelength. And up above red is infrared. That is heat. Has an even longer wavelength. Microwave radiation has an even longer wavelength, and radio waves are much longer, like up to 1,000 meters long. Down on the violet side, it's a short wavelength, 380 nanometers. Below that, the part we can't see, is UV radiation, down to 10 nanometers. Then X-rays, and then gamma rays are the shortest wavelength. So pigments are going to be able to absorb some of these wavelengths of light, some of the spectrum, but others they will reflect. What our eye is seeing is the wavelengths that bounce off, that are reflected. Leaves are green and plants are green because chlorophyll A and chlorophyll B are absorbing blue and yellow wavelengths of light and red wavelengths of light, and they are reflecting the green. That G of Roy G. Biv is what's being reflected. So they're not using those wavelengths. But we see those wavelengths. If you have a black shirt on, it absorbs all wavelengths of light. That's why they're hot. White doesn't absorb very much because it's all is reflected, and we see that as white when it's all mixed like that. When electrons are absorbed, they're boosted to higher energy levels, and then the wavelength of light that a pigment absorbs depends on the amount of energy needed to boost an electron up to another orbital. So just like, it has the orbitals. First is there's the 1s orbital, and then there's the 2p, and there's 3 2p or propeller-shaped orbitals, and then one 2s orbital. Those orbitals, electrons can be pushed out of their orbital. And if they're boosted up to a higher orbital, that takes energy. Okay, different pigments allow plants to absorb different wavelengths of light, and that in being able to absorb a number of different wavelengths of light, they're able to take the most advantage of that spectrum, of that energy. So once it absorbs energy, an electron absorbs energy, it's boosted up here to a higher orbital, to the second shell. It's in an excited state, and it's unstable, and it releases the energy as heat or light. And those excited electrons can be transferred over to an electron transport chain or captured in another molecule. Chlorophyll A and B each contain this porphyrin ring. Here's chlorophyll A, and here's beta carotene over here. And in chlorophyll A, there's a CH3 hanging off of this porphyrin ring. And in chlorophyll B, it's CHO. But that porphyrin ring has a little bit of magnesium sitting in the middle of it. And beta carotene, a little bit of magnesium. And that little magnesium matters. Beta carotene has 2 smaller rings. And this is a chart that shows the different wavelengths of light that are absorbed by chlorophyll A, chlorophyll B, and beta carotene. We're going to try looking at these when we do an extraction and we do paper chromatography with the leaf pigments. So as you can see, this shows the wavelengths on the x-axis from 350 to about 700. So we're going from violet or indigo, I guess violet, all the way to the red spectrum. And then the relative absorption of light at the wavelength is up on the, that's funny, it says on the x-axis, but it's actually up on the y-axis. That shows how much absorption occurs. So you can see, due to these pigments, beta carotene absorbs quite a lot of the light at the blue, green, and violet spectrum, actually more blue and violet spectrum, beta carotene. Chlorophyll B absorbs blue light, chlorophyll A more violet light. And then down on the other end of the spectrum, chlorophyll B is absorbing some at the red, and chlorophyll A even lower at the red or infrared, and not much is being absorbed here at the green at 550. This mirrors, the action spectrum, which is the rate of photosynthesis by a whole plant at certain wavelengths. So the rate of photosynthesis drops dramatically at 550 nanometers, the wavelength of green light, and increases very dramatically at about 425 nanometers, which is the wavelength where chlorophyll A and chlorophyll B are really able to absorb a lot of light. And also down here at about 675 nanometers. Okay, so these pigments are absorbing light. They're reflecting the green light, but they're absorbing the other wavelengths of light. And what can happen is, as they absorb light, it excites the pigment, it excites electrons in the pigment, and those electrons will bounce to another molecule and produce energy intermediates for work in the cell. In the thylakoid membranes of chloroplasts, there are complexes of molecules. The first one that was discovered was, I always say this differently, photosystem 1. Photosystem 1, but there's another one that happens before photosystem 1 that it took us a while to figure out. And since we discovered it secondly, it's called photosystem 2, but photosystem 2 happens first. Remember that little trick. Photosystem 2 happens first. In both of these photosystems, light is exciting a pigment and boosting the energy level of electrons. Okay. So it happens first in photosystem 2. It's the first step. Light hits the pigment and it bounces electrons out of the pigment. And in the process also in photosystem 2, water gets split. A water molecule gets split or oxidized, and oxygen is produced. It's produced right there at the beginning of this process. Oxygen is produced in photosystem 2. Hydrogen ions are produced, and as happens so often in these energy production steps, just like in cellular respiration, there's an electrochemical gradient that is produced because of the hydrogen ions that is produced across this electron transport chain. The electron transport chain, electrons bounce down that chain. Those little electrons bounce down that chain, and as they bounce down the chain, they energize these proteins, transmembrane proteins, to pump hydrogen ions into the inner lumen of the thylakoid. Okay, there's an electrochemical gradient that's set up. These electrons are bouncing down that electron transport chain, and they meet photosystem 1, where their photosystem 1 is making has NADP, NANP there. And sweet NANP is accepting those little electrons home, NADP. And when she accepts those little electrons, they become NADPH. And the NADPH helps to suck up hydrogen ions from the stroma that the thylakoids are sitting in. Okay, so in the chloroplast, once again, ATP gets generated by chemiosmosis. that is now, instead of calling it oxidative phosphorylation, because we're not using oxygen here, right? We produce oxygen when water gets split. They're at the very beginning of photosystem 2, but we're not really, that's a waste product. That gets breathed out of the stomata. So what we're using is NADP. And what they're calling this is photophosphorylation. Photophosphorylation. It's taking a phosphate group and attaching it to ADP so that it becomes ATP. And the whole process is driven by the flow of hydrogen ions through that cool circus ride. It's the coolest protein I think there may be. And you've got a lot of cool proteins in you. This one's not even in you, it's in plants. But you have it in you in your mitochondria. It's the same one that we dealt with in cellular respiration called ATP synthase. Cool circus ride of a protein that spins and produces ATP. This hydrogen gradient across that thylakoid membrane is generated in 3 ways. Water is split, so that increases the hydrogen ions in the thylakoid lumen. In the electron transport chain, hydrogen ions are pumped into the lumen by the electrons bouncing down those proteins of the electron transport chain. And when NADPH is formed, it sucks up some hydrogen in the stroma outside of the lumen. And so it creates this difference in hydrogen ions across that thylakoid membrane. So this shows photosystem 2. First is photosystem 2. Light is hitting this pigment that's called the light harvesting complex. It can be chlorophyll A and B or carotene. But then it gets those little electrons all excited, that light does, and they're so excited that water gets split. Water gets split. They're so excited they gotta pee immediately, and water is split. And oxygen is produced. But then the hydrogens in water actually contribute then to this electrochemical gradient. Lots of hydrogen ions or hydride ions or protons being pumped into the thylakoid lumen, inside the thylakoid. So it goes photosystem 2, these electrons start bouncing around and they bounce on down and they start bouncing down the electron transport chain. They get to, oh my goodness, they go they get transferred to a pigment called P680. That's where the oxygen's created. They bounce on down to quinones and other kinds of embedded proteins and down this cytochrome complex. And each time, hydrogen ions are pumped into the lumen. Then they get down to photosystem 1. And photosystem 1 has another boost of light that helps to boost those electrons, and that's collected by a pigment. And those electrons, oh, fortunately there is sweet NADP there, NADP, is there at the end of the electron transport chain as part of photosystem 1. She's there to accept those electrons home. And so as NADP gets made into NADPH, that soaks up some more hydrogen ions that are outside of the thylakoid in the stroma. Well, what are all those little hydrogen ions going to do? What do any good little hydrogen ions do, or hydride ions or protons? They go through a ATP synthase, cool little circus ride of a molecule, and a whole lot of ATP gets generated as that thing spins. It's the only way that those hydrogen ions can get through and equalize the concentrations on either side of that thylakoid membrane. Okay, so what's produced from the light reactions? Oxygen's produced, and it's really a waste product. And 2 electrons get transferred to P680 molecules. And then NADPH is produced in photosystem 2. NADPH is produced in photosystem 1. Photosystem 1 is the second one. So NADP plus joins with electrons and hydrogen to become NADPH. So nana-P is there at the end to get those electrons. And then ATP is produced, a lot of it. This time it's photophosphorylation, because oxygen isn't used. But ATP synthase spins because of that electrochemical gradient of hydrogen or hydride ions, and it phosphorylates a lot of ADP into ATP. So this is a linear process, right? It's really a non-cyclic process. Electrons start, the whole thing starts at photosystem 2, and eventually they get to photosystem 1 and NADPH. It's linear. And it produces ATP and NADPH in equal amounts. But then there's also a cyclic photophosphorylation, where this electron cycling releases energy into the lumen, and only ATP is produced. And so, and that is a cyclic photophosphorylation that happens. So this is the cyclic photophosphorylation, and actually there's a feedback mechanism here where if there's a great deal of energy produced, if there's a lot of NADH produced, it's going to go back and shut that off. So these complexes in the electron transport chains are very similar between chloroplasts and mitochondria, showing really the relatedness of everything. These genes are derived from a common ancestor, which is so cool. Cytochrome B proteins will play a very similar role in both mitochondria and chloroplasts. So very similar genes used. And this shows these homologous proteins. Here's cytochrome B, F, and this is cytochrome B, C, and they both have these very similar proteins, cytochrome B proteins. Okay, photosystem 2 has the light harvesting complex, which includes the pigment, right? And it's also called the antenna complex, and it absorbs the photons. And then the energy gets transferred. And then there's a reaction center. This reaction center is not very stable. Energy is transferred very quickly, and this is where water is split or broken up, and oxygen is produced there in the reaction center. And so the oxygen gas is released as waste. Okay, here's photosystem 2. Light energy gets absorbed by a pigment, like chlorophyll A or B. The energy is transferred among the pigment. It's kind of spread out because of resonance energy until it reaches P680. And that P680 gets converted. There's a high energy electron on it. And it's very stable, but then it's going to bounce an electron out onto the electron transport chain. So the energy curve has a zigzag shape or a Z scheme. So because there are decreases and there are increases and then corresponding decreases in the energy of an electron. So if we're just looking at the energy of the electron, that shows the Z shape. So here you have these electrons that are energized here to the primary electron acceptor from P680, and then the energy starts to go downhill, and then they gets boosted again at photosystem 1. And then it starts to go downhill until NADPH welcomes those little electrons home. Goodness, I'm getting sleepy. I shouldn't have had my lunch before I did this. Okay, I'm going to stop this here, and we'll get to the Calvin cycle when we gather next time. Take care, and I hope you have a great weekend.